the dinucleotide duplex. So I'll probably give you the structures of all the nucleotides, and then you have to put them together into a dinucleotide and then match that up with another dinucleotide to draw a proper hydrogen bonded duplex. And the others will probably give you one or two chemical descriptors of fatty acids, and then we'll just have you draw out the chemical structure. Yes?
Regarding the midterms, does that mean that every exam will be worth 50% of the final grade? If you adjust the scale, if you drop an exam?
That's correct.
Okay.
That's correct. Yeah.
Whose laptop is this?
Does that change my email regarding? Does not change that at all. We will still be working with a very heavy curve. Yes. Yeah, the remaining 10% will come from the recitation. That's, whether or not you've been actually showing up and your performance on things like the quizzes and like those problems that we'll do in recitation next week. That will be the remaining 10%. If you can call it a quiz. Yeah. All right. Yes.
Regarding the quiz, would Monday also officially still have it this upcoming Monday?
Yes. And we will send out some additional details of exactly what you should be expected to do in recitation. That will be included within the homework assignments for next week. And we will provide real nice details in there. Good. Yes. There are, as far as I know, no recitations the week of Thanksgiving. Maybe an optional something for the Monday people. I don't think we need to get caught up with the recitation, so I think there's just no recitations, but I will update you on that. Yeah, certainly no recitations Wednesday, Thursday, Friday. Any other items? Good. All right. So we went through some basic aspects about the history of DNA structure and how we knew that it was a functional molecule. And I want to stress again that it's important for you to know how we know these things, not just like, okay, I know A binds to T and G binds to C, but just knowing how we know some of these basic aspects of DNA structure and function is pretty important. Just a quick reminder, once again, what we think about when we look at biological macromolecules, we think about the monomer units, we think about their chemical properties, we think about how they are built up, bonded to each other to form a polymer chain, and once they're present in a polymer chain, the structural attributes of those polymers. So here are our monomer units for DNA. And once again, the full monomer that's included in the nucleic acid structures includes the phosphate group, and we call those the nucleotides. But I want to make sure you don't get confused with the nucleosides, which are just the ribose and the base. And we emphasized last time the differences between DNA and RNA structure, and again, just showing the four monomers that are used to build up RNA. So I should also mention that there are some quirky kind of bases that get inserted into nucleic acids. Okay, so we have the canonical ones, AT, GC, and U. But occasionally we'll see some weirdos show up, not so much in DNA, but very often in RNA, and especially in tRNA, transfer RNA. The most common modification is like a methylation, like we have right there, or right there, with a guanosine or adenosine. And when we talk about where those methylations are, if we're just providing a methylation right onto the ring, we just provide the ring number, so this is 5-methylcytidine. And if we're talking about methylation of an exocyclic position, what do I mean about exocyclic? I mean this nitrogen is methylated. It's not part of the nucleobase heterocycle. It's off the ring. It's exocyclic. And so in those cases, we get the atom type and ring number. So it's N2, that's the position, N2 methyl, and then base, that's guanosine, so that's N2 methyl guanosine. All right. Some other weirdos, inosine is a variation that pops up very often, especially in the anticodon of tRNAs, and we'll see why when we go to decode mRNAs. And then here's another little interesting one. This is pseudouridine. This actually does not have the base conjugated through a glycosidic linkage to that nitrogen. Instead, that ribose is conjugated at the C5 position there. Okay? So you don't need to memorize these by any means, but you need to be familiar with them, especially because we're going to be seeing instances in which methylation of DNA serves as an epigenetic mark, which we will focus on a little bit later in the semester. All right. So the other variation is not just in the nucleobase, but it's in the position of the phosphate. Typically, we write these things as phosphate groups on the 5' position, but we can find that phosphate wandering around a little bit. Sometimes we will observe it dangling off the 3' position, the adenosine 3' monophosphate, or it can dangle off the 2' position, and then we sometimes see this cyclic monophosphate structure, 2', 3' cyclic monophosphate, and we get these guys typically as a result of hydrolysis of RNA. And you'll see in just a bit where these are coming from. So this not only shows up as just something that happens when you degrade RNA, but we talked about cyclic AMP before as an important second messenger when we were talking about biosignaling. So this plays another role as far as signaling is concerned. All right. So we've got our monomers. We're going to build them up into polymer chains. And I'm just going to provide the chemical description of that today. And then next week, we'll talk about the enzymatic action that begins to build up DNA from a DNA template using enzyme-mediated polymerization. Yes?
Briefly, in the previous slide, the camp that was shown is 2,3. Is there a difference between 2,3 and 5,3, or?
Yes. There is a difference between 2,3 cyclic and 5,3 cyclic. Right? So if you were to make the ester linkage from here down to here, that would be the 5,3. This one is 2,3. So those are different structures with different properties, different functions. Yeah. Good. Okay, so here's our chemical description of what the polymer chains look like. And we make our linkages. They're phosphodiester linkages. Right? So polypeptides, the linkages are amide groups between the monomers. These are phosphodiester linkages. And these form part of the backbone. The position, yes, it matters where these ester linkages are positioned. We make the linkage from the 3' hydroxyl of one nucleotide to the 5' hydroxyl of the next one. And so if you want to think about the backbone, it goes back and forth, ribose phosphodiester, ribose phosphodiester, ribose phosphodiester. That's the composition of the backbone. Both of those groups are polar, right? Certainly the phosphate group is bearing a negative charge. For sure that's polar. And the ribose is a polar saccharide as well. The phosphates are charged. The pKa is low. You should not write your phosphates in an uncharged form, as Linus Pauling mistakenly did. And that means that this is a polyanionic polymer, and there needs to be counter ions to balance those negative charges. And those counter ions, like typically in the form of, let's say, magnesium for RNA structures, is really important when we take a look at the three-dimensional structure of well-folded nucleic acids. All right, so when we were talking about polypeptides as polymers, we talked about the amide groups constitute the backbone, and that the side chains were the 20 different functional groups present in the 20 different natural amino acids. And in this case, our side chain groups are just the 4 bases for DNA and the 4 bases for RNA. This extension of the polymer backbone is directional. We're taking a 3' hydroxyl and we're associating it in a covalent linkage through a phosphodiester with the 5' hydroxyl. This is directional, and we have distinct ends. This is the 5' end, this is the 3' end. So it's not a symmetric molecule, and they have distinct features at either end. And I mentioned this last time, when we write out the structure, we write it out from the 5' end to the 3' end. So this would be ATGCA and not ACGTA. Okay. Good. Questions about the nature of the DNA polymer? All right, let's bust it apart. We talked about this last time because we talked about the difference between RNA having this 2' hydroxyl and catalyzing the hydrolysis of the phosphodiester linkage right at the neighboring position. And I'm showing this to you again just to indicate to you where this 2', 3' cyclic monophosphate structure comes from. And then if you were to break one or the other of the linkage here in this cyclic structure, you get a mixture of 2' and 3' monophosphate derivatives. So that's where those structures typically come from. All right, so that's great. We've got our individual nucleic acid strands, but now we need to build up not just the primary structure, but the secondary structure, and in the case of RNA, the tertiary structure as well. So how are we going to organize these individual chains, polymer chains? In the case of DNA, Watson and Crick's big breakthrough was that we're going to form a duplex. We're going to form a 2-stranded helix. And what defines the way in which each of these strands comes together is the fact that there can be hydrogen bonding between the purines and the pyrimidines to make these AT and GC base pairs. That defines the nature of the interior of the double helical structure. And so we call these base pairs. And actually, I'm not too happy with what I wrote here, that the helix could be stabilized by hydrogen bonding. We'll talk a little bit more about this during the course of this lecture, but the question is, does hydrogen bonding define the specificity with which these strands come together, or does it really drive the stability? Where does the stability come from for forming a DNA duplex? So maybe the original idea that occurred to Watson and Crick is, yeah, we're forming hydrogen bonds, big energetic payoff. That's what's stabilizing the structure. And I'm going to give you another possible interpretation as we go through the lecture today. But if you have 2 strands and you're trying to figure out, okay, where are they going to come together? How are we going to line them up? For sure, these hydrogen bonding interactions mediate specificity in how we form these duplexes. Because if we insist that an adenine always pairs with a thymine and a guanine always pairs with a cytosine, that will really establish how these 2 strands are going to come together. So that was really one of the big breakthroughs for Watson and Crick, is that they identified this complementarity. They noted right away that A forms 2 hydrogen bonds with T, and 3 hydrogen bonds are formed in the GC base pair. And the big thing that they realized as they assembled their structure, it's like, okay, not only does this avoid these preposterous chemical features that other people had suggested before, including packing the phosphates in the interior of the helix, but it actually explains so much more. It explains char gas rules, right? Why did the purines always equal the pyrimidine concentration, regardless of what species they were looking at? This explains it. If you always need to pair up a purine with a pyrimidine, this is going to explain char gas rules. And the other thing is, it immediately established a mechanism for duplication. And as I told you last time, there was all this evidence that had accumulated that DNA was the transforming factor, right? That you could provide DNA to a bacterium and transform it, that it was supplying information, and that it was presumably the genetic material that would require this information to be duplicated from one generation to the other. What's the molecular mechanism for that duplication? Base pairing. So here is the model that Watson and Crick established. It is a double helix. It's a right-handed double helix. Doesn't have to be that way. This is one of the forms of DNA. Turns out in this form it's right-handed. We'll see another example. The backbone is hydrophilic. The interior is hydrophobic. We like that. The bases are stacked in the interior, and they're oriented perpendicular to the axis of the helix. Okay? So they're stacked one on top of the other. As we form the double helix, there's going to be regions which allow access towards the interior of the helix, and it actually allows molecules from the interior to contact portions of the base pairs. And there's 2 grooves that allow that kind of access. We call the larger one the major groove. The smaller one is the minor groove. And here's an important feature that I should have emphasized before. As we line up the 2 strands to form the hydrogen bonds between them, the base pairs, they are anti-parallel. One goes from 5' to 3' in one direction, and the other goes in the opposite direction, 5' to 3'. Okay? Anti-parallel. Questions about that? All right. Here is another depiction of the B form structure of DNA. This is now a space-filling representation for all these atoms. And now you can begin to see quite clearly, all right, here is our major groove right there, and our minor groove is there. Just barely see it between the phosphodiester backbone. So you can see these gray spheres in there actually represent atoms that are associated with the base pairs. And it becomes obvious, like, okay, yes, the base pairs are somewhat sequestered, but if you really wanted to interact with them, there is this opportunity to reach in through the major groove. Bases are packed so that they have very good van der Waals interactions between them. About 3.4 angstroms between the plane of one base pair and the next. About 10.5 base pairs per helical turn. And that, you multiply those 2 together, you get 1 full turn of this B-form DNA gives you 36 angstroms. Okay, so I was saying before about stabilizing forces. Okay, yeah, easy to see the hydrogen bonds, but base stacking also is going to be really important, because as you pile these base pairs one on top of the other, really good van der Waals contacts. There's probably kind of aromatic stabilizing interactions, maybe some dipole interactions as well, and also this forms a sequestered hydrophobic surface area. So it's probably this base stacking that's really providing the bulk of the energy for forming these DNA duplexes. And specificity, for sure, is being established by hydrogen bonding. That base stacking is fairly non-specific, but it seems to provide a very strong driving force. Okay. Just to emphasize this, we do have strand complementarity. So if I were to pull these strands apart, right, we have plenty of information here on both of these strands, but if I was to pull one of these strands apart, and if I was to throw it away, I could completely duplicate the original structure, because all the information that I need to recreate this structure is contained within that one strand. All I need to do is line up the proper base pairing partner on one of these strands. I can pick either one to be the template. I just lay down the monomers, stitch them together, and then I can reform this duplex. So the sequence of one strand uniquely determines the sequence of the other strand. And that really is a nice aspect that immediately occurred to Watson and Crick when they published their famous paper in 1953 in Nature. And if you look really carefully, towards the end of this paper, right here, there's this amazing understatement, which is one of the biggest understatements that I've ever read in any scientific report whatsoever, which is, it's almost droll. It's like, it has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. Like, of course it does. I mean, that was the really big breakthrough with the structure that they proposed. Once you had the bases in the interior of a duplex form, right? I mean, yeah, it was appealing to put the nucleobases on the outside for reasons we talked about last time. But once you have that complementarity, as I said, you pull one strand apart, that's the template for resynthesis of the original material. And you can do that on both strands, and then you can make two copies of your original genetic material, and you can provide the original genetic information to both cells upon cell division, and that allows you to copy genetic material, genetic information from one generation to the next. Huge immediate advantage in the structure that they proposed. For those of you who are kind of interested in this historical thing about why Linus Pauling came up with the wrong structure, why he was in such a rush, there was some discussion about, I mean, the guy won 2 Nobel Prizes, right? He was no dummy. Why did he come up with this incorrect structure? What else was going on politically? It was, he was being harassed for his leftist political views, and he was having trouble traveling out of the country, getting visas. There's a fascinating history, and it's maintained at the Oregon State University Library, so if you're interested in that history, it's available through that link. The other kind of unfortunate, I guess, aspect about the history of this solving the structure of DNA is the recognition or lack thereof that was accorded to Rosalind Franklin. And as I showed you before, she was a chemist and a crystallographer. She obtained the X-ray diffraction patterns of the wet form, hydrated form of DNA that really showed so clearly the helical structure. She passed away not too far after her publication of her crystallographic data. It was published in the same journal as the Watson and Crick paper, but it seemed as though Watson and Crick weren't really eager to give Rosalind Franklin the real recognition for her contribution. And actually, you know what, I'm going to turn back to this paper and just point out that if you go back and look at the 1953 Nature paper, I mean, typically when we take a look at real seminal, groundbreaking scientific articles, you see this compelling data set, because the greater the claims of a scientific discovery, the more substantive the data set needs to be. The data that Watson and Crick provided basically all came from Rosalind Franklin. I'm not saying that they didn't do a lot of work. Their imagination, their understanding of chemical principles, chemical bonding, figuring out how these base pairs were lined up, the anti-parallel orientation of the strands, all of which was fascinating and insightful. But as far as the data that those models were built on, it all came from Rosalind Franklin. And it would be perhaps more tolerable if this wasn't a pattern of behavior, but unfortunately it was kind of the tendency at the time, and also it's probably indicative, unfortunately, of the kind of person that James Watson at least was. And he's actually still associated with a research center out on Long Island. He's quite elderly, but he, as he got older, felt quite comfortable not only in diminishing Rosalind Franklin's individual contribution to his discovery, along with Crick, but also kind of making chauvinistic comments in general. And he was, I think, recognized that people had difficulty with his outspoken character. He knew that people found him to be quite unbearable, but perhaps it wasn't just while he was young. Maybe he got more outspoken in a very unpleasant way as he became older. And fairly recently, he made some horrible sexist and racist remarks. I'm not going to provide them to you now. If you want to find them out, I'm sure they're available to you. I find it really disheartening because I look to these people like Linus Pauling, Rosalind Franklin, Watson Crick, and these others as like role models. I mean, I'd love to be making those kinds of discoveries. And it's just so disappointing when people who've made these breakthroughs and have recognized for their scientific talent feel as though it gives them the platform to make these sorts of outrageous statements about things that they have no expertise whatsoever, such as the racial basis of intelligence. So for me, you know, smart, famous people, they're not always right. Pauling in particular, who I think is a complete genius, he was wrong all the time. And just because you're clever doesn't make you somebody that other people should aspire to as a role model. Just because you win a Nobel Prize doesn't make you a worthy human being. It doesn't make you a visionary in all human knowledge. And just because you're wrong doesn't mean that you can't be a decent person as well. So I'm, just throwing that out there. It would be difficult for me to laud the accomplishments of Watson and Crick scientifically, which I certainly do, without making these additional comments. It's a little bit more difficult for me, maybe, because very early on, after I got to NYU, I got an award. I applied for, scientific recognition. It came with some research funds. And the name of the award is the James D. Watson Investigator Award. And at the time, I was like, oh, wow, Watson, Crick, Watson. And now I look at it, I just go, So later on after class, I don't want to take up your time. I'm going to try to sand his name off of my board. See how that goes. I'll scrub him from my CV too. All right. So let's talk about this B-form DNA. Where does the minor and major group come from? So here's our base pairs for AT and GC, and you see in both cases there's a much wider opening into that base pair on one side than the other. And yeah, we can actually read off what those base pairs are. If we put in a small enough probe, we could see a pattern of hydrogen bonds that would be distinct within that major group, right? But the B-form helix is just one of the secondary structures that DNA can form. I like this representation of it because it makes it clear what's distinct about B-form DNA than others. We're looking right down the helix here, and you can see that the interior of the helix is really kind of filled up. There's not very much empty space. And if we draw this representation here with the base pairs as rectangles, we see that the center of the base pair is aligned precisely with the center of the helical axis. And yeah, again, the planes of these rectangles are perpendicular to the axis. Why do we get to draw the base pairs as these rectangles? I mean, AT is not the same, A is not the same as T, G is not the same as C, AT not the same as GC, but here we're just drawing everything the same as a rectangle. And we can get away with this because it turns out that as we make those base pairs, the shape of an AT base pair is pretty close to the same shape as the GC base pair. So even though the individual bases got different sizes, the base pairs themselves are about the same. And that explains why we don't see huge variations in the overall geometry or of the structure as we vary from a GC-rich to an AT-rich sequence. It looks pretty much the same in one portion of the molecule to the other. All right, but not the only form of DNA. After the initial discovery of Watson and Crick's double helical form, it was realized that there was this A form of DNA. It's pretty close, and you can probably interconvert from one to the other. You could probably populate forms that are somewhat intermediary between one and the other. We more often see A-form DNA when we have RNA present, right? And we can get that by forming a duplex between DNA and RNA or forming two strands of RNA and allowing them to come together. It's a bit more compact in that there's 11 bases per turn, not 10 and a half. And in this case, in the A form, the bases are slightly tilted away from being perpendicular to the axis of the helix. So they're a little bit twisted, tilted up. And then here's the big difference as far as I'm concerned. Here's the way that the base pairs are arranged. They kind of wander around the exterior of the interior of the helix. So that leaves a little bit of space right in the middle. We mentioned last time that the ribose rings in DNA versus RNA, they have a different kind of pucker. And in this case, in A form DNA, we see a C3' endo form, which is distinct for the A form structure. Okay. So just to compare the B form and A form DNA, here's our B form. Base pairs more oriented right over the middle of the helix axis, and the A form DNA, those base pairs wander around the outside. And yeah, the rise per residue, the distance from one to the next is slightly larger for the B form DNA than the A form. And then here's what you can see with regard to the twist, the tilt of the individual base pairs, right? So the helix axis runs up and down, and the base pairs are tilted slightly away from that, being perpendicular to that helix axis. I want to just mention that nucleic acids have their own dynamics, and we talked before about the dynamic features of proteins. They're not static structures. Phi inside dihedral angles can vary. They exhibit motions. And we did talk about, what bonds are rotatable, right? The amide bond, not so much because of its partial double bond character. Phi inside dihedral angles, yes, those are variable. Those can exhibit dynamics, and the side chains certainly can exhibit motion as well. There is substantial conformational heterogeneity. There's motion in DNA, and we have a large number of rotatable bonds per every nucleotide. There's you can count 6.5 rotatable bonds. You can see them right here. This fourth one within the ribose ring itself, it can undergo slight motions, right? But there's no way that you could spin that particular bond around 180 degrees. That's constrained within the ribose ring itself. So slight motions around that carbon-carbon bond within the ribose. We mentioned before the different puckering within the sugar, C2', C3', endo, exo, and then rotation around bond 7 shown here. That is the glycosidic linkage to the base itself. Okay, so let me give you a better view of what that rotation around that glycosidic linkage looks like. These form two different types of conformers, the syn and the anti. Okay, so here's our motion around that glycosidic linkage from the ribose carbon to the heterocycle nitrogen, base nitrogen. And you can see that you can swing, in the case of the purines, you can swing the bulk of the purine heterocycle over the ribose. That's the syn conformer. Or you can swing it away, and that's the anti. And then with regard to the pyrimidines, it's really, we're looking at that carbonyl oxygen, whether or not it's over the ribose ring in the syn conformation or swung out in the anti conformation. So those are the sorts of motions that nucleic acids can exhibit. Okay, so we've got A form, B form DNA. They're populated a little bit differently depending upon their hydration state, and that's why it was so crucial that Rosalind Franklin was working with well-hydrated DNA so that she drove her DNA sample completely into B form. If that wasn't controlled properly, you'd get a mixture of A form and B form DNA, and the X-ray diffraction patterns that you would get would be kind of confusing and difficult to work from. Yeah.
Which of these conformations are preferred?
It can depend upon the form of DNA that you're dealing with. So we'll get into that right now. So in Z form DNA, this is a third form of DNA secondary structure. We have a left-handed helix. Both A form, B form DNA are right-handed helices. This is a left-handed helix. Here, once again, is this little kind of way to look at helical structures where you point your thumb down the helix axis and then wrap your fingers around it. And this is going around towards the left, right? So left-hand DNA. This was really thought to be a curiosity, like, okay, if you take DNA and you torture it and you place it in forms that would never experience within the cell physiologically, okay, you can really mess it up and get this thing to completely rotate from a right-handed form to a left-handed form, but it's just a complete anomaly. This guy named Alexander Rich, who was one of the very first nucleic acid chemists, he was really interested in whether or not there was a physiological role for this. It's still quite mysterious, but there's some evidence now that it actually may play a role within the cell. It's restricted to DNA. RNA doesn't do this. It does have a C2' endo, and in this form, we populate syn forms for the purines and anti-forms for the pyrimidines. So, syn forms for the purines, where they're rotated over the base, and anti-forms for the pyrimidines. So here was a structure that Alexander Rich contributed to that just came out not that long ago. And this kind of blew people's minds. Yeah.
Can you explain one more time why hydrating the DNA drove it all to the Z form?
I can't explain what it is about hydration that favors B-form DNA over A-form DNA, but that's been a consistent result. As you dehydrate DNA, it tends to prefer A-form. That's a good question. If you find out more about it, let me know. Okay. All right, so seems like the Z-form DNA might not be so much of an oddity. They actually were able to crystallize a section of DNA that contained both Z-form DNA and B-form DNA. And here's what that looks like. So here's my question to you. Where's the Z-form? Where's the B-form? They're both there. Can you spot where they are and where the boundary is between them? Keep in mind, Z-form is weird because it's got a left-handed helix, and those gray lines are charting out the phosphodiester backbone. The backbone is charted out in those gray lines. Which part has the zDNA? zDNA is at the bottom. Great. So let's look at these gray lines here. They're rotating around towards the left. And at the top, it looks as though they're twisting in the right-handed conformation. And in fact, that to me looks like a pretty normal minor groove of DNA, B-form DNA. So there's the juncture right there. And that's pretty amazing. We've taken DNA, left-handed, 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 all of a sudden, boom, it switches over to right-handed B-form DNA. How does it do that? It arranges for that structural swap by kicking out two bases. Here's a thymine over here, an adenine over on the right-hand side. They've been pushed out from the interior of the helix in order to accommodate the switch in backbone structure. So this was really cool. And now people began to say, oh, okay, well, if Z-form DNA and B-form DNA can coexist, then maybe there is a physiological role. And it turns out that there are proteins that stabilize that Z-form DNA, and that's actually how they got this piece of nucleic acid to crystallize in that form. They co-crystallized it with the protein that interacts with the Z-form DNA. You can see down here in blue. This is a nice structure just showing right here and here where the bases have been extruded from the duplex. I also like the fact that this paper pointed out that as much base stacking as possible was accommodated within the structure. There's only two bases, one on each side, that are kicked out, and everywhere else it's maximized the amount of base stacking interactions, and they conclude that this confirms that base stacking constitutes a major stabilizing factor. And so this gets back to this question about, is it hydrogen bonding or base stacking that's really driving the stability of DNA? All right, yeah.
Why isn't that just
Oh yeah, I mean, for sure that would be a major complaint. Like, just because you were able to isolate this one piece of DNA, put it in a crystallization apparatus, have huge amounts of this stabilizing protein present, and then you're not talking about this in the context of like chromosomal DNA. You're like swapping it out with this protein that's like bending it into this particular form. I think that's a fair complaint that this might still not be really indicative of what's going on physiologically. So that's why I think it's still somewhat controversial about the real role of zDNA within the cell. All right, so here are the three forms: A form, B form, Z form. Right-handed helices for A and B. You can see that left-handed helical form for Z. And then again, A form is more present in low water environments. The helix is wider. You can see it's a little bit more opened up. And the minor groove is a little bit shallower. And then only the Z form DNA has the syn orientation of the bases over the ribose, and only for the purines. All right. Questions about double helical DNA structures? Good. All right. As we begin to think about how to organize DNA, if we just propagated these helices and we had all like 3 billion bases of DNA in each one of our cells, we would be dealing with a structure that would be way too long. So we need to compact DNA in order to tuck it away within our nuclei. And we're not going to get into too much detail about chromatin structure right now, just to say that the double helical forms do become wound around themselves, and they do form higher order tertiary structures. So if you were to take, let's say, a circular piece of plasmid DNA, you could wind it around itself, and then you could create these supercoils that are shown here. And as you create these forms, you can induce different topoisomers, right? That represents how many times one of the strands gets wrapped around the other. And these differences in topological forms turn out to be really important when it comes to replicating DNA. And we'll take a look at enzymes that are used to interconvert these different topological forms of DNA. Show you a little bit more about these topological forms. So we can take a circular piece of DNA and we can twist it around itself either in one direction or the other. These correspond to what are called positive supercoils or negative supercoils. We can undergo conversion between these different forms by cutting one of the double-stranded DNA and passing another strand through it. And so we can interconvert between different topological forms. And this just shows electron photomicrographs of what a piece of circular DNA looks like without any supercoiling. We call that relaxed, and with some negative supercoiling. So yeah, this compaction turns out to be really important. We also want to alleviate some of the electrostatic issues with regard to packing DNA. And so within the nucleus, DNA gets wrapped around histone proteins. And again, we'll get back to this a little bit more when we talk about DNA replication. But this is a really nice way to condense DNA. We take very positively charged proteins. They're going to be complementary to the negatively charged DNA backbone, and then we wrap the DNA around those protein bulk. And as we do that, it's in a particular orientation. It does induce negative supercoiling around that histone because of the controlled orientation. That negative supercoiling is compensated for positive supercoiling in other portions of the DNA structure. And then again, if we were to use an enzyme to cleave the DNA double strand and allow it to relax, we would just be left with a negative supercoiling of the DNA around the histone. And that's what we see when we take a look, electron photomicrographs of DNA in the context of its chromatin structure. We see these kinds of beads on a string of DNA wrapped around what are called these nucleosome particles composed of these positively charged histone proteins. Just to give you a little bit of an intuition for the complexity of the topological issue, if the cell didn't have some way to control topological forms, you'd probably end up with a topological disaster like these phone cords that we used to deal with when I was using phones in an earlier era. And then the cell really tries to tuck the DNA away very nicely and control topological issues by packing the DNA around these histones. And then again, I'm going to emphasize this when we get to DNA replication, but the topological issues become really critical when you take two of the strands and you pull them apart. And you need to do that because you need to get access to the bases in order to replicate them. Right? All the pictures we've been showing, like, oh yeah, we can copy the information from one strand to the other. Those bases are in the middle of the double helix. In order to gain access to them, you need to separate the strands. And this is exactly what happens during DNA replication, during DNA templated RNA polymerization. The strands need to get separated. What happens when you do that? If you go to a particular place and say, I'm going to start my replication here, I'm going to pull the two strands apart, behind that, what we call that transcription bubble, we're going to be generating negative supercoiling, that's topological stress. Ahead of the transcription bubble, we generate positive supercoiling. And if we were just to keep doing this and doing this and doing this, as we move down the DNA, replicating it or translating it, we would end up with a really horrible situation, and things would come to a screaming stop because there would be a very high energy form as we began to populate these topologically stressed molecules. And again, how does the cell overcome these issues? Enzymes. Enzymes are brought in to alleviate this stress, and there are a family of enzymes called topoisomerases that travel down the DNA, cleaving the DNA and alleviating the supercoiling issues. And again, we'll get into that with more detail when we talk about replication. Okay, other unusual forms of DNA. Talk all the time about double-stranded DNA. We mock Linus Pauling for coming up with a triple-stranded form. Doesn't he know? In fact, you can get triple-stranded DNA. You can take a canonical double-stranded form, like an AU base pair here, or a GC base pair over here, and you can line up an additional base and get hydrogen bond complementarity between them. As I pointed out, you can reach in to the major groove and you can contact hydrogen bond partners. So this adenine is reaching in and it's finding a hydrogen bond donor and a hydrogen bond acceptor on that AU base pair, and it's saying, okay, I'm going to tuck adenine side chain right in there to make this triple-stranded form. Again, you can tuck a guanine into a GC base pair in the simple way. Another really quirky one is a guanine tetraplex. So we can line up guanines to make this unusual hydrogen bond complemented form. All four of them come together and create this roughly square-shaped arrangement. And if we were to take these four guanines and then repeat that motif over and over and over again, we create these G quadruplex stacks. And obviously the only way you're going to be able to do that is if you have a region of DNA that's very rich in guanines. But we see these. These so-called Hoogsteen base pairing, these triple-stranded forms, you will find them in RNA structures in particular. And they end up, populating sometimes extended forms, even in DNA, where you can get these triple helical arrangements. It's really only RNA that forms more complex globular structures, and this just shows some of the high-resolution structures that have been solved for RNAs as they begin to populate bulges, hairpins, these sorts of things. This is hammerhead form of RNA. This is, I believe, catalytic. This is a portion of RNA from a virus, and this is a portion of RNA that forms an intron that needs to be cleaved out as RNA is being matured. We had this huge explosion of information